Welcome back, everyone. Next, we have Diakonos Oncology, Inc., a recipient of FDA's Fast Track designation. It's a clinical stage biotech company developing a groundbreaking dendritic cell vaccine for difficult to treat cancers. Please welcome its CFO and Head of Business Development, Anthony Baldor. Nice to see you, Anthony.
Thank you, Anna. Good afternoon. Good morning. I'm Anthony Baldor, CFO and Head of Business Development for Diakonos Oncology. Thrilled to share with you the new frontier in cancer vaccines. So we are cracking the code on harnessing dendritic cells to fight cancer. So we'll start with a little bit of background. Companies developing a highly differentiated double-loaded dendritic cell therapy that was discovered at MD Anderson and then further developed at Baylor. With this approach, we've achieved clinical success in some of the most challenging cancers, including glioblastoma and pancreatic cancer, among others. I'll cover some of the data later in this presentation, but just to grab your attention now, one key takeaway is the 12-month survival rate in our ongoing phase 1 glioblastoma study, which is currently at 91%. And you'd expect that to be closer to 50 or 60%. So none of the four cohorts has reached median overall survival, and nor has the full study. So those are great. These early data, survival data, as well as an excellent safety profile, have enabled us to pursue an aggressive regulatory strategy. So we received FDA Fast Track designation, as Anna mentioned, for the glioblastoma program in October of last year, and we received Fast Track for the PDAC program just last month. So the GBM program is also awarded Orphan Drug designation in January of this year. We're running a lean team with low overhead. Companies headquartered in Houston, including leadership, R&D, and manufacturing. We have key personnel, finance and business development in the Bay Area biotech cluster, and regulatory in Silver Spring, Maryland, nearby the FDA. Let's see, a couple more vital stats at the outset here. We finished raising an $11 million seed round earlier this year, which should fund the company into mid-2025. We expect to start our phase 2 glioblastoma study in the fourth quarter of this year, and the Series A is designed to cover those costs. Finally, we do have 3 active phase 1 studies. The technology really should work in any tumor type, so there's an immense opportunity here. And I'll just highlight to that end, critical portfolio strategy work is ongoing. So to take a traditional approach to the pitch, what's the problem that we solve? There's a massive unmet medical need in glioblastoma that I will talk about in a minute. But as I just highlighted, the opportunity here is much bigger than glioblastoma. So appreciating there's a mixed audience here of biotech and generalist investors. I'll skip the immunology lesson, but just give you a few bullets on the history of cell therapy that hopefully will resonate with everyone. Most of you will be familiar with CAR T-cell therapies. These products have become huge successes for treating blood cancers, sending many patients into complete remission. Top-selling product, Yescarta, is generating $1.5 billion in revenue annually. But engineering T-cells was the backup plan. T-cells are educated by another white blood cell type called the dendritic cell. These cells are master regulators of the adaptive immune system, capable of mounting a complete immune response through a number of subordinate cell types, including CD8 and CD4 T-cells. They can broadly target the full array of tumor-associated antigens, which is really important for heterogeneous tumors like glioblastoma. And that's where the industry started its cell therapy fight against cancer. The first public clinical studies for dendritic cells was more than 10 years before the first clinical CAR T publication. So it's not controversial to say dendritic cells should work, but drug developers have failed to really harness their power. And history teaches us that priming a dendritic cell with tumor proteins only or tumor mRNA only is insufficient to cause the dendritic cell to drive an effective Th1 cytotoxic cancer-killing immune response. This is illustrated in the chart on the bottom right with the Th1 activation marker, interferon gamma. So staying on the chart, if you prime a dendritic cell with both tumor mRNA and tumor protein simultaneously, it responds with an order of magnitude greater Th1 cell-killing activation. So this is the big deal. Diakonos co-founders have cracked the code, it seems, on dendritic cells. And they did it with a simple solution, with an elegant solution that mirrors what a viral infection looks like to the dendritic cell. And a virus is the infection type that, like a tumor, naturally requires an immune response where the immune system kills cells. So really only in a viral infection will the dendritic cell encounter antigens found externally from viral particles floating around outside the cell, and internally from viral mRNA inside the infected cell itself. In that situation, the dendritic cell is double loaded by nature, and the dendritic cell seems to take this very personally. So that was the core discovery that set Diakonos in motion. And the teams followed up to show that dendritic cells actually compare mRNA and protein antigens internally inside themselves when they're determining what kind of an immune response to elicit. So I won't cover much of it today, but there's a lot of strong science that supports and differentiates this technology from other cancer vaccines, other dendritic cell approaches. But if that's interesting and you want to know more, we'd be very happy to go deeper. Very quickly, before I move on, just to be clear on the double loading concept, the cartoon in the bottom left, to double load is to prime the dendritic cell with both tumor proteins and tumor RNA separately and simultaneously. So hopefully that's clear. This slide seeks to put some data behind the claims I just made on the previous slide. So the gene expression analysis illustrates two things. Firstly, the double-loaded dendritic cell responds in an entirely different way than a dendritic cell that's single-loaded with RNA only. So protein only, or even if you falsely double-load with unrelated RNA and proteins from different sources, the cell will not respond in this way. Secondly, the gene expression changes and signaling pathways that are activated or deactivated in response to that double loading. One, strongly upregulate that Th1 cell killing capability, and two, downregulate the alternative Th2 pathway. And the team knows an immense amount about this signaling pathway and how and why it works. So again, the team is happy to go deeper on the science because it does help explain why those clinical data are looking very good right now. Okay, so we defined double loading, but just to go a layer deeper on the full procedure, so we're all on the same page. So step one on the left, starting with the person icon at the bottom, we take a tumor sample, and in glioblastoma, the surgical resection is conveniently part of the standard of care. From that sample, we process, we're on step 1B now. From that sample, we process the tumor into protein and mRNA. And I'll highlight that preparing protein plus RNA not only enables that full double loading activation, but it also helps capture a fuller spectrum of tumor antigens than if you were to use only one source or the other. Okay, 2 quick points before we move on to step 2. Unlike with CAR T and other leading cell therapy approaches, this process does not require any genetic modification. So that translates into vastly simpler and less costly manufacturing, which is a key challenge facing CAR T developers today. The second is RNA is pretty finicky. So there's this and a couple other aspects contribute to a very strong IP portfolio around how to effectively prep the tumor and execute the double loading. Okay, step 2. So back on the person icon on the bottom, we then collect the patient's white blood cells through apheresis, step 2A. These are treated to give us dendritic cells, which are then double loaded separately but simultaneously with tumor protein and RNA. Step 3, dendritic cells compare those proteins, RNA antigens become, and they become 10x activated through that robust and unique set of signaling pathways that I shared on the previous slide. And those cells are then injected into the patient near their lymph nodes, cervical lymph nodes, which are the draining lymph nodes for the brain, which is a strategic route of administration. So the lymph nodes being the location in the body where dendritic cells meet T cells to activate that full adaptive response and train those T cells how to kill the tumor. Okay, the other problem we're solving, glioblastoma is a significant opportunity. With the standard of care, which is surgical resection, chemotherapy, and radiation, the expected survival is less than 15 months for all glioblastoma patients. overall, and the chemo is not believed to really help a large subset of patients, which are described as unmethylated patients. So for those patients, the survival is much lower. 5-year overall survival, under 10%. So big, large unmet need, tragic disease here. For Diakonos founders, glioblastoma was attractive because there's a very strong scientific rationale for tackling this tumor type with a dendritic cell vaccine. I mentioned already how the dendritic cell vaccine tackles the full mutation and antigen spectrum of a tumor. And GBM is a highly heterogeneous tumor type. So many past attempts to treat GBM are believed to have failed because they only address a single target. We call this targeted therapy. It works very well for some tumor types, but not for GBM. I also mentioned the tumor sample that we need to prep the vaccine is collected as part of the standard of care. So supporting rationale there. This can be a very large market. There are 25,000 new cases of glioblastoma diagnosed annually in the 7 major pharmaceutical markets, and nearly 15,000 of those are in the United States. Without suggesting a price for Doc 1021, we're still seeing very high pricing power for cell therapies. Without guiding a peak sales estimate, a conservative P times Q, price times quantity, can easily get you to $1 billion of annual revenue. And yes, CARTA illustrates this is achievable for a much more complex product than we're proposing. Market research not complete, but a few things that we know today that I'll share. There remains high willingness to pay for oncology and rare diseases. Iovance recently priced their cell therapy Amtagvi at over $500,000 for a course of treatment. I described the low COGS and simple manufacturing relative to other cell therapies. And this is, I think, an interesting one. If we can give these patients 3, 4, 5 years of life, but their cancer does eventually come back with a new set of mutations, there's no reason to believe we wouldn't be able to redo this procedure and provide the same benefit against the new tumor and the new antigen set. So potential for redosing. Okay. We have treated now 18 glioblastoma patients in a phase 1 study. These swim lanes represent the 16 newly diagnosed patients. The other 2 were suffering from recurrent disease, which is a different prognosis. There are a few key takeaways for these data. 12-month survival is currently at 91%, and that's for the 70% of patients with enough follow-up to be evaluable. So a majority of those patients. None of the 4 cohorts has reached median overall survival, nor has the study in full reached median overall survival. Median follow-up is currently at 13 months, with 75% of patients still alive, and good performance status scores at the last report. So we're optimistic that they'll continue to survive for some time. These outcomes are highly statistically significant. Or these current data are highly statistically significant when compared to matched historical controls by Kaplan-Meier analysis with a p less than 0.003. We're optimistic. We're really encouraged by the current data. We're going to continue to track these patients, through the end of the year and beyond, hopefully. But before I move on, color coding. I mentioned earlier the difference between the easier methylated patients and the more difficult unmethylated patients. We have only 1 patient with a less challenging methylated disease. That patient's identified in gray. Rest of the patients are the more challenging unmethylated subtype, which makes, I think, these data even more impressive. Many other GBM clinical trials will enroll up to 60% even of their study patients with a better prognosis methylated type. So this is a very challenging patient population we're treating. Additionally, in the dark green, we highlight the unmethylated patients with additional poor prognostic indicators. So aspects of their demographics or disease that make their expected outcomes even worse. These are patients who would have been excluded from other clinical studies, either because their disease progressed between the surgical resection and the vaccine administration. Maybe they could not receive a full tumor resection due to its location in the brain. Maybe their age. Most studies won't take patients older than 65 or 70. We took several, or some combination of these issues. So the founders accepted these patients into the phase 1 study because they believed in the technology, and 2, they're in pursuit of the mission to cure cancer. They said yes. So arguably a risk from a drug developer's point of view, but that choice is helping us today because the expected survival for these poor prognostic patients is really estimated to be closer to 8 months for the dark green bars. Okay, I'll speed up a little bit from here. Earlier in the pitch, I covered the foundational science that supports the great data story. These next few slides offer additional reasons to believe from a clinical biomarker perspective. Are the DOC1021 dendritic cells doing what we expect? Is there a measurable change in the immune response before and after vaccination? Is a, quote, full immune response, including immunologic memory, initiated? And these data suggest that yes, treatment with DOC1021 induces robust immunologic memory. So these charts depict pre and post-vaccination increases in central memory CD8 T cells, central memory CD4 T cells, and a pronounced increase in CD127 cells. So CD127 being sort of less commonly known as a cell marker, sort of a bona fide long-term effector memory T cells. GBM tumors are naturally behind the blood-brain barrier. And there's a debate about how intact the blood-brain barrier is in patients with brain tumors. But there's some evidence the barrier may be more intact than previously hoped. And this would help explain why past failures for certain biologic drugs that have been studied in glioblastoma. That said, because dendritic cells do their work to train the immune system in the lymph nodes, our drug product, DOC1021, does not need to cross the blood-brain barrier. Only its effect on the immune system needs to reach the tumors to put on the fight. And dendritic cell-trained T cells would be far more dynamic, capable of trafficking themselves to a tumor than in sort of an innate protein is. So we've got a couple patients who had additional surgeries after they were treated with DOC1021. And these patients' tissues confirm pretty striking before and after infiltration of the remaining tumor area by cytotoxic cell-killing CD8 T cells, which are stained in red. This may be slightly harder to see, but there's also reduced tumor cell density in the after versus the before. Those are the little blue cells. And so this is highly encouraging biomarker for us. Top images are before and the bottom images are after, if that's not clear. Okay, so that's promising clinical data with the robust underlying science and confirmatory biomarkers to support the story. Let's talk about safety. This is a phase 1 study, and DOC1021 has shown itself to be highly tolerable. No DLTs, no dose-related toxicities. No related grade 4 adverse events, only 3 grade 3 adverse events, possibly or probably related to treatment. And then not described here are some common treatment-related grade 1 AEs, which were mostly flu-like symptoms and injection site reactions. And I'll note that flu-like symptoms are in a way positive because they suggest that there's an ongoing immune response. Okay, great. So on that backdrop, we are designing a phase 2 study, which we hope to initiate by the end of the year. This is in the final stages of design, so I'm going to discuss only some of the high-level parameters that are more settled. The first is this will be a head-to-head study of DOC1021 given in conjunction with the standard of care compared to the standard of care alone. And this will help justify future use as part of first-time standard of care therapy. We believe a proper control like this will be required by potential commercial partners or for the FDA to consider accelerated approval in this indication. The second is based on the treatment effect we're seeing in the phase 1, we're targeting a 120 patient study with 60 patients receiving DOC1021. The full study size could shrink if we don't do a one-to-one randomization. The third is we're targeting 15 sites to enroll and execute the study expeditiously. And I'll share, we've been thrilled with the interest to date. We've got 14 sites under CDA and in active discussions. This has been not only a little surprising, the yield, but it's also highly validating that these experts, these people who are treating glioblastoma patients day in and day out, and hearing this story and seeing our data, much more data than I have been able to share with you today. They're excited to work with this drug candidate. So that's a thrill. Endpoints, the FDA has made it pretty clear that they want a survival endpoint for the primary. How that's measured is yet to be determined. We have several options. We're exploring options also for progression-free survival as a secondary endpoint. And while the FDA values that a little bit differently than partners and investors, progression-free survival will provide an opportunity for interim data reads on changes in tumor size, response rate, et cetera. The investment community really values those. So, of course, we'll continue to track safety. And for the clinicians in the audience, the simple flow chart on the right takes that diagram of the treatment process that I showed earlier in the presentation and puts it on a timeline for you, schema. Okay, so what else do we need in place to start the study in Q4? We need GMP manufacturing established, and this is well underway. We announced our partnership with Cellepont in October of last year. The facility is 76,000 square feet in the Woodlands, Texas, near the Houston headquarters, but also in close proximity to George Bush Intercontinental Airport. They'll be ready. Tech transfer is complete. Gap assessments complete. Potency and release testing are in development, and additional process development, including automation, are also underway. And we recently brought on a new head of CMC, which adds to the operational progress here that really complements the maturing clinical data. The team, very quickly, CEO Mike Wick's serial entrepreneur with several large exits to his track record. Recently added Laura, CMO, Laura Consulting CMO, Laura Aguilar, is a perfect fit, really. She's co-founder at cancer vaccine company Candel Therapeutics, and before that, 10 years at Dana-Farber Cancer Institute. Another recent addition, Paul Lammers, brings biotech leadership, substantial cell therapy experience to the board. He's been an invaluable add. Head of regulatory, Ann Belair, spent over a decade as a regulatory insider at the FDA. I'm your CFO, currently head of BD, over 2 decades in biotech finance, strategy, and business development. I joined the company earlier this year to support the Series A raise. COO, Jay Hartenbach, another highly successful entrepreneur, part of the angel funding group that set Diakonos up to close the seed financing, which he led. Chief Scientific Advisor Will Decker led the founding scientific team, was the PI for our phase 1 studies, currently professor of immunology at Baylor College of Medicine. And we'll highlight here 2 key members of the scientific advisory board, Drs. Tandon and Curry, an accomplished neurosurgeon and neuro-oncologist. Okay, in summary, Diakonos is developing a highly differentiated double-loaded dendritic cell therapy. We have fast track designation awarded for both glioblastoma and PDAC. Doc 1021 showing strong safety and efficacy in the phase 1 glioblastoma study, and dosing is complete for that study. Phase 2 GBM study is targeted to start by the end of the year. Scalable GMP manufacturing will be ready by phase 2 in the 4th quarter. We've got a strong and growing team in IP portfolio, and we're seeking $30 million to fund that phase 2 GBM through clinical proof of concept. Thank you. So Anna, I think we have time for some questions.
Yes, we do. Thank you for that. So if this technology can be used in any cancer, how will you prioritize which tumor types to focus on after GBM?
Yeah, this is a great question. So I mentioned we've got a lot of pipeline and portfolio strategy work ongoing. In terms of first principles, right, that guide our decision making, we're looking at expected efficacy based on other immunotherapy results in that indication and setting, ability to combine with a standard of care in earlier lines of treatment, tumors that are easy to access for material needed to make the personalized vaccine, a population that's large enough to be of interest from a market point of view, unmet medical need, fast pathway to approval, and then sort of a favorable competitive landscape that's going to either help our development and or permit us to be part of the race.
Talk a little bit more about differentiation. How is this approach different from other cancer vaccines out there?
Yeah, another great question. So I think key takeaways here are, compared to other cancer vaccines, our approach is a dendritic cell vaccine, which, as I mentioned, can initiate a full adaptive immune response, address the full mutation and antigen spectrum in a tumor. This is going to be critical in glioblastoma. So single target approaches, whether EGFR, EGFRV3, survivin, IL-13, receptor alpha 2, et cetera, there's plenty of targets. They're going to encounter challenges if they can't kind of attack a more full spectrum. compared to other dendritic cell vaccines, our double loading mRNA plus protein is pretty differentiated, protected by strong IP, supported by robust underlying science. That's sort of what this presentation has been about. This is a very different approach, and we're seeing what looks like a very different outcome. So this also kind of differentiates us from the Moderna approach. Moderna's cancer vaccine is not a dendritic cell vaccine, but we get the question a lot because the company is very well known. They're providing a spectrum of antigens, but in the end, they have to choose which ones. And so we allow nature to find the best antigens from the full tumor profile. The Moderna approach only provides one antigen source for the dendritic cell, and that's not going to enable double loading. So there's reasonable differentiation there as well. When compared to CAR T approaches, dendritic cells initiate a full and natural immune response versus the CAR T single agent engineered artificial response. CAR T approach does have to pick a single agent, maybe two in some cases, but it's severely limited from a target heterogeneity point of view. And CAR T approach, because it's sort of artificial, tends to have worse safety outcomes than we see with dendritic cells. The last point is compared to other treatments for glioblastoma, the multi-target full immune response mechanism will be required, for success in this indication. So that's a key takeaway from connecting with key opinion leaders earlier this year. Single target approaches have not worked to date, and they're not expected to start working in this heterogeneous tumor type. And then also we know that immune activation is going to be required for treatment success. So we see this even with simpler technologies like chemo. When they do rarely work, they tend to provoke immune responses in those patients. And immuno-oncology approach like Doc1021 is the way.
Perfect. And this is a great question to close. Talk a little bit about some milestones and future fundraising.
Yeah, perfect. So the $30 million Series A is designed to cover the proposed phase 2 only, with a 10% cushion on already conservative CRO budget. In this certainly thawing, but still challenging capital market, we're first looking for a well-scoped investment that can significantly de-risk the technology with that formal clinical proof of concept. As the interim phase 2 data emerges and hopefully starts to confirm the great phase 1 results, and as we align on the ideal pipeline programs, we can look to raise a round to support development beyond GBM. Whether that's in 2 or 3 years will be determined by markets and data outcomes. And then additionally, with the phase 2 top line data currently expected in 2027, we would seek to find a commercialization partner, or we can consider an equity round to cover confirmatory phase 3. A lot of options out there. And then, of course, we have a number of non-dilutive grant funding options that we're currently working on, or planning to apply for.
Perfect. Well, thank you so much, Anthony. We appreciate your time and thorough presentation. We'd love to have you back in the future with some updates.
Very good. Thanks, Anna. Appreciate the opportunity.
All right, everyone, stay with us. We'll be right back.